go. Yeah, so afternoon and welcome everyone. We are pleased to have you join us for our first installment of the Sharing Ocean Acidification Resources for Communicators and Educators webinar series of 2018. Source is presented by the NOAA Ocean Acidification Program and National Marine Sanctuaries in partnership with Washington State Sea Grant and the Institute for Systems Biology. Our goal is to provide ocean acidification communication tools to formal and informal educators and stakeholders across the country to promote a more integrated and effective ocean acidification education community. I'm Jen Bennett-Mintz, Education and Outreach Coordinator with NOAA's Ocean Acidification Program, and I'll be moderating today's session. We also have Laura Francis, Education Coordinator from the Channel Islands National Marine Sanctuary online, and she will be behind the scenes today helping answer questions from you all. We have had close to 150 registrants for today's webinar from all over the US and Canada, including Alaska, Washington, California, Maine, New Hampshire, Georgia, Alabama, Florida, and many places in between. Thank you all for being here today. We are glad to have quite a diverse group in several different time zones getting accustomed to that extra hour of daylight in the evenings. So during the presentation, all participants will be in listen-only mode. You're welcome to type questions related to technical issues or questions for our presenter into the questions box in the bottom of the control panel on the right-hand side of your screen. We'll be monitoring incoming questions and we'll respond to them or pose them to our speaker at the end of her presentation. And we always welcome your input on what source webinar speakers you'd like to see or topics or resources we cover in upcoming webinars. So if you have ideas for us, please type them into the questions box and we will take those into consideration as we plan for the future. We are recording this session and we'll share the recording with registered participants via Google Drive and on the NOAA Ocean Acidifications Program's YouTube channel. So the title of today's presentation, Why I Put a Pteropod in a CT Scanner to Study the Impacts of Ocean Acidification. And our speaker today is Rosie Oakes. We are very pleased to have Rosie with us. She's currently at the National Academy of Sciences of Drexel University. Rosie is a geologist by training and stumbled into the wonderful world of pteropods after finding some shells in a sediment core she was working on during her PhD. Since then, Rosie has spent over 200 hours CT scanning pteropods and has used a variety of other imaging techniques to learn more about how these organisms may be affected by ocean acidification. Rosie believes that it's important to communicate science on all levels, and so in addition to traveling to international science conferences and publishing scientific papers, she makes time to attend school science fairs and participate in outreach events, just like this one, and really hopes to inspire that next generation of scientists. Rosie is originally from the UK and is currently living in Philadelphia and working as a postdoc research fellow at the National Academy of Sciences. So I've had the great pleasure of meeting Rosie in person, and I'm really looking forward to hearing more, not only about her research, but these really neat 3D tools she's using for outreach. And with that, Rosie, I'm going to pass it over to you.
Okay, thank you very much, Jen, and thank you very much everybody for coming along today. I'm really excited to be doing this webinar and to tell you guys a little bit about my research and how I've used that research in outreach. So you can see the title slide right now. You can see some pteropods, which are going to be the focus of the talk today. On the left of your screen, you see a live pteropod. You can see its beautiful, pristine, glassy shell as it's swimming through the ocean. And then the next photo across is an X-ray of a pteropod, and then a 3D reconstruction and a shell thickness map of a pteropod, which are all things that we are going to learn about today. So before we get started, I'm just curious who we have listening today. So I think Jen or Laura are going to post a question on your screen. And if you could fill in the poll and answer and let me know where you guys are coming from today. And then I think Jen will give me the feedback so I know who I'm talking to.
Yes, Rosie. So everyone hopefully is seeing a poll on their screen. You can go ahead and just click right on the screen to answer, whether you're a research scientist, teacher, informal or experiential educator. Maybe you classify as a science communicator or other. And if you are in that other role, we'd love for you to just type in what that is in the questions box so Rosie can get a sense of who's on the line. All right. So we'll leave the poll open. Rosie, it looks like we have a great variety on the line that are falling into that other category: aquaculture farmer, a representative from the macaw tribe in the Pacific Northwest, oceanographers.
All right, well, that sounds great. Well, thank you all for tuning in, however you found out about the webinar. All right, so let's get started. I'm gonna, oh, all right. Oh, Jen, I think your microphone is still on.
I will turn that off. And just so you know, we do have about a quarter that are in the research science field. A little less than that are teachers, a third are informal educators, about a fifth are science communicators, and then you've heard some of those others on the line.
That's great. Thank you very much. All right, well, we'll get going now. So just wanted to start off by giving you an outline of what I'm going to talk about today. Firstly, I'm going to introduce why I'm interested in pteropods, and then why I'm studying the pteropods. I mean, they're a really interesting species to study, but how I got into this personally. Then I'm going to talk about why I use CT scanning and what I've learned using CT scanning and how we learned this, which probably is the most interesting research-wise. And then how you can use this information that we gain from the CT scanner to help with science communication and pteropods. And finally, I'll finish off by talking a little bit about what is next for me and my research. So why pteropods? So for those of you who don't know, pteropods are planktonic mollusks. You can see the images here. They're pretty diverse, different shaped shells, but essentially their name comes from ptero, which means winged. You might have heard of a pterodactyl. It's a pair of wings, and poda, which means foot. So they have a winged foot, which they use to swim through the oceans. And you can see the shape varies a lot. So in the top left corner, you have Limacina helicina, and the top right corner, you have Limacina retroversa. These are both polar species. And then in the center, that's a Steelina sabata. So they're found throughout the world's oceans, and they're a really diverse group. But these things are really tiny. So in the bottom corner, you can see a picture of my finger next to pteropods, which are mounted for CT scanning. They're about the size of a grain of sugar. So they're really tiny, but they're really important in the ocean. And they do migrate through the water column daily, which is pretty cool. So they are down at about 200 or 300 meters depth during the day, and then they migrate up to the surface at nighttime. So I just have a video here of the pteropods swimming using those wings. You can see that actually their nickname is sea butterflies, and they swim the same as butterflies. They get both propulsion from both the forward and the backward stroke of their wings. So you can see how they propel themselves through the oceans. So why are pteropods at risk? So this is a graph of atmospheric carbon dioxide levels measured at Mauna Loa in Hawaii from 1960 to the present. And you can see that over the past 80 years, there's been an 80 part per million increase in carbon dioxide. So this is really rapid. And the effects of this we're seeing in changing global temperatures, changes in storm patterns, and changes in heat and stuff in different parts of the country. But I'm interested in a secondary effect of increasing carbon dioxide, and that's the effect that it has on the oceans. So since the Industrial Revolution, it's predicted that about 30% of the carbon dioxide which has been emitted into the atmosphere has been absorbed by the oceans. So if that process hadn't been occurring, that graph on the previous slide would look even steeper than it already did. So this carbon dioxide dissolves into the ocean and reacts with water, forming carbonic acid. This acid then dissociates to form hydrogen ions and bicarbonate ions, and some of these hydrogen ions then react with carbonate ions in the seawater, forming more bicarbonate ions. So carbonate chemistry can be a little bit complicated, but essentially what you need to take away from this slide is that there is a net increase in the amount of in the water column. So increased hydrogen ions is a decreased pH, so the ocean is becoming more acidic, and hence the name ocean acidification. And there's a net decrease in the concentration of carbonate ions in the seawater. Now, carbonate ions are what calcareous organisms or organisms which build their shells from carbonate use to build their shells. They like little building blocks for those shells. So it makes it energetically less favorable for them to build those shells when you decrease the concentration of carbonate ions. So before we go any further, I just want to go through some terminology so that we're all on the same page moving forward. So pH, as I mentioned previously, is a measure of acidity. The more hydrogen ions there are, the lower the pH is, or the more acidic it is. Now, one really important thing to point out is that the pH of the modern ocean is 8.1, so it's actually alkaline. And if we add more hydrogen ions, it's going to become more acidic, but it probably won't go actually acidic. It will still be alkaline. So that's something really important, and something that the term ocean acidification can be a little bit confusing. Next, we're talking about the concentration of carbonate ions in seawater. And these, as I said previously, are used to make carbonate minerals. Now, there are two forms of calcium carbonate. There's calcite and there's aragonite. So organisms like mussels use calcite to build their shells, and organisms like pteropods use aragonite to build their shells. So chemically, these two things are absolutely identical, just the way that those different atoms are stacked within the chemical structure are different. And the result of this is that aragonite is actually 1.5 times more soluble than calcite. And so, although they're both chemically the same, organisms which make their shells from aragonite are at higher risk from acidification than those which build their shells out of calcite. And the final term we're going to talk about today is saturation, or omega aragonite. And this looks complicated because it's a formula and it can be a little scary, but essentially it's just the ratio of calcium and carbonate, the concentrations of calcium and carbonate in seawater divided by the concentrations of calcium and carbonate at equilibrium for that mineral. So at equilibrium for the formation of aragonite. So if the concentration in seawater is higher than that you need at equilibrium to form aragonite, then the water is supersaturated with respect to aragonite. That value is above 1. However, if the concentration in seawater is less than you need at equilibrium, the water is undersaturated, and that means that the mineral aragonite is then chemically unstable. Okay, so I'm done with all the terminology now. So hopefully you'll remember these things moving forward. So using this new term that we've just learned, omega aragonite, this is a global surface ocean map of omega aragonite from 1850 to 1860, with undersaturated values below 1 are in red, and the supersaturated values from 1 to 3 are in orange, yellow, and then blue. And the first thing that you notice is that the ocean is not a bathtub. This water is not the same everywhere. The chemistry of this water is not the same globally. And the biggest trend that you see is that saturation at the poles is lower and saturation at low latitudes is higher. And one of the causes of this is that gases are more soluble in cold water. Think of your nice cold fizzy beer versus your warm flat beer. The cold fizzy beer is holding more gas. And in the case of carbon dioxide, it means you're holding more carbon dioxide and more of that is dissolving into the water, lowering the saturation. This map here is a projection for surface aragonite saturation for 2100. And the first thing you notice is that the pattern is much the same as the previous map that we looked at. However, there's been a global shift to lower omega aragonite saturations, and you can see that now polar and subpolar regions are experiencing undersaturated waters. And this is obviously a massive change that we're seeing over this time period, and it's really going to affect the organisms that live in these areas. And one of these organisms are the pteropods, as I talked about previously. This species, well, all the pteropods form their shells from aragonite, which is that more soluble version of calcium carbonate. And these species in particular live in polar and subpolar regions. And so they've kind of got the double hit for ocean acidification. They're living in the worst place and they have the worst type of shell. And so for this reason, pteropods have been considered kind of an indicator species for ocean acidification, or the canary in the coal mine for ocean acidification. So why does it matter anyway? Why does it matter if we lose this group of sugar grain sized swimming mollusks from the ocean? Well, it turns out pteropods are delicious. They are the potato chips of the sea. So they're really high in lipid content and things love to eat them. So you can see here the organisms that we have that feed on pteropods are the gymnosperms, which are the shellless pteropods or sea angels. They don't look particularly angelic, but they feed exclusively on pteropods. And then krill and myctophid fish also feed on pteropods. And these groups of organisms are fed on by penguins, whales, and seabirds. So pteropods actually play a really key role in ecosystems, especially in polar regions. So it's really important that we understand how they're impacted by projected future changes in ocean chemistry. So why me? How did I stumble into studying this species? Well, as Jen mentioned in the introduction, I'm originally from the UK, near a town called Chester. This is the village where my parents live. And growing up, I was really interested in how landscapes form. It's like why the mountains were a certain shape or why that valley was there. And so I decided to go to the University of Edinburgh to study physical geography and geology. And while I was there, I got interested in these big global cycles of nutrients, of elements, and how that affected all the other organisms. So I actually moved to the University of Toronto to do my master's, where I studied how phosphorus cycling in the Cretaceous, which was about 100 million years ago, like how changes in the phosphorus cycling impacted the organisms that lived in the ocean. So then for my PhD, I wanted to move somewhere where I could focus more on learning about the biota itself from these fossil records. So I moved to Penn State University. But as Jen mentioned, through all this time, I was a geologist. So this is a picture of me in the field at the end of my second year, where I'd been digging through trenches in the desert in Utah, looking for ammonites, which were big ancient mollusks that were around in the Cretaceous. And I was interested in how they were impacted by changes in the climate in the past. So that's what I was doing with my time, being a geologist, looking at past ocean chemistry changes. And it was actually while looking at some cores which were taken from the Cariaco Basin, which is just off Venezuela, I was looking at how changes in ocean chemistry over the past 20,000 years had impacted ocean biota. I found some pteropods, and I actually really didn't know a lot about them. I hadn't seen them in any of the samples that I was looking at before. And as geologists, what we usually do when we find this is we look to the modern. So we think, right, I really want to know what was going on with these organisms in the past. What can these shells tell us about what the ocean was like 20,000 years ago? So we think, okay, what do we know about them in the modern, about how nutrients affect their size or their thickness, or how carbonate chemistry affects their shells, and what can that tell us about the past? However, when I looked to the literature, I found that this stuff hadn't actually been studied for pteropods in the modern. So in an effort to understand the past, I thought, oh, I'll quickly just go into the modern for a little bit and see if I can figure some things out. But as ever with science, I'm still here. I've got more questions than I have answers. So I still don't know what was happening with them in the past, but first it's important that we understand what's happening with them in the modern ocean. So how on earth did I come to CT scanning these organisms? Well, traditionally, pteropods have been studied in 2 dimensions. So researchers would put pteropods underneath the light microscope and look at their shells. So as you saw in the title slide, pteropods' shells when they're swimming in the modern ocean are beautiful. They are pristine, they're glassy and transparent. But when they dissolve, they're visibly affected by that dissolution. So they turn from this lovely glassy color into kind of a milky white color. And then with further dissolution, as you can see in kind of number 3 up here, they lose, they become opaque, and then they kind of get pitting and they lose their sheen. So people have used this kind of as a scale of how dissolved is a sample is how bad do the pteropods look. And this study is actually looking at pteropods from the fossil record, looking at, you know, how much dissolution has occurred in these samples. And the advantage of this is that using a light microscope is really quick, it's easy, and it's cheap. So you can process large numbers of samples relatively quickly. However, the negatives of this are that you're only getting a qualitative measurement. You're looking at these shells and you're saying, this is a 0, this is a 4, this is a 2. And it doesn't really measure exactly the change that's occurring in the pteropod. With the light microscope, you can only see the outside of the shell. So you're only seeing one aspect of the change which is occurring. And with this study, the aim was to look at changes in the preservation of sediments over time. And so you're looking at big changes in preservation. But if we're expecting to look at changes which might occur in pteropod shells linked to ocean acidification, those changes are likely to be much, much smaller. Maybe a slight thinning of the shell or a small area of dissolution. It's very unlikely that the organism would have a shell that looked like number 5, for example, and would still be alive. So people have tackled this by looking at pteropods under the scanning electron microscope. So this is a study from Lischka and Ribosel in 2012, and so a scanning electron microscope uses a beam of electrons instead of light as the source for the microscope. So you can see things in much higher resolution. So you can see here they have a pristine shell, and they have a shell which has undergone some dissolution. So the advantage of this is you can see in higher resolution than at the light microscope. However, the scales that these are assessed on were still qualitative. So people were still ranking how dissolved a shell was on a scale of 1 to 3 or 1 to 5, depending on researchers. So the advantage of CT scanning is that you add another dimension to the study. So instead of looking at things in 2 dimensions, you're then looking at things in 3 dimensions. So this is the CT scanner that I used. It's made by GE, and it's a micro CT scanner. And they use it for all sorts of cool things, like looking at airplane engine parts or looking at the insides of your iPhone, scanning them to see if there are any defects. But it can also be used for scanning plankton, which is pretty exciting. And they work much like a medical CT scanner. So in a medical CT scanner, you would lie down on the bed portion of this, and then the X-ray and the detector would rotate around you, taking a lot of images from lots of different angles. And from those images, you can then reconstruct a 3-dimensional view of whatever it is you were looking at. So in this example, it's a 3-dimensional reconstruction of the head and neck area. So CT scanning a pteropod works much the same way, except you mount the pteropod on a glass rod and the pteropod rotates rather than the whole machine rotating around the pteropod. So you get an X-ray tube, and you get a beam of X-rays coming out of that tube. And when the X-rays interact with the shell, if there's material there, it will slow down the X-rays or partially block the X-rays. So then there's much less reaching the detector. So we put the pteropod in the CT scanner, and it rotates around on this glass rod like this. And then that's the end of the scan. So it's question time again. And I think Jen is going to put the question up there. So I'm interested, how long do you think it takes to CT scan 1 pteropod shell? I can only put 1 in at a time because their shells are so thin. You have to get very high resolution scans. So I think you're going to see 4 options on your poll. So it's either 0 to 15 minutes, 15 to 30 minutes, 30 to 45 minutes, or 45 to 60 minutes. So have a little think, and I think Jen will report back to me once you've had a guess of how long it takes.
Got it, Rosie. Yeah, the guesses are coming in, so we'll collect those responses for just a few more moments. All right, Rosie, I don't know if you can see the responses.
I don't think so.
All right, it looks like almost half of the folks on the line are guessing between 15 and 30 minutes, and about evenly parsed out with those other options.
Okay, great. Thank you very much. Well, I wish it was that quick. Sadly, it takes 52 minutes to scan a pteropod, so quite a long time. So 52 minutes to scan one pteropod shell. And the reason that it takes so long is because, as I mentioned previously, these shells are so thin that we're really close to the detection limit of the CT scanner itself. So hopefully, as CT technology develops further, we can shorten the scan times. So we take 1,000 images per scan. And as I said, it takes 52 minutes, and we get about 2 gigabytes of data for every shell that we scan. So big data sets that we're working with, but you can do really cool things with the data that you get. So from the scans, we make 3-dimensional reconstructions of the shells. So from these reconstructions, you can measure things such as thickness or shell volume. And we're trying to do some shape analysis as well to see if the shapes of pteropods change. So the advantages of CT scanning is that you can look at these organisms in high resolution, and you can make those quantitative measurements which you couldn't make with the 2D imaging techniques that I talked about previously. You can see both internal and external features of the shell, which is really interesting, and I'll talk you through in my case study example how that's really helped us learn some more about the pteropods. But obviously the negatives of this technique are that it is expensive and it's time consuming. So you can't look at very many in terms of the number of specimens that you look at. You're really limited compared to the light microscope. So eventually, I assume what will happen in terms of monitoring these pteropods moving forward is that we'll use a combination of light microscopy to look at the pteropods in bulk, and then we'll CT scan some of those specimens. So that would be my prediction at this point. But obviously, there's nothing laid down at the moment. So now I'm going to talk you through a case study about what we've learned about pteropods by CT scanning them and how we learn this. So just to recap, the problem is that pteropods are at risk from ocean acidification because they make their shells from aragonite, which is the more soluble form of calcium carbonate. And the species that I'm interested in, the Limacina helicina in this case, reach their highest abundances in polar regions, where aragonite saturation state is expected to become undersaturated the earliest. So they're going to be the first to be impacted. So this is a cross-section through a shell. So pteropods are gastropods. So all gastropods have a protective organic covering on the outside of their shell. So you can see in the top left corner, this is just a normal shell. This is to give you an idea of what this diagram is of. So this part here, the mantle, that's like the body of the pteropod and the wings that you saw previously. And then this area here labeled S is the shell. So then on the outside of the shell, there's this thin organic layer called the periostracum. And it forms as the shell forms. The shell is actually laid down against the periostracum, but it's subject to decay and abrasion. So if something tries to predate this shell, or it's scratched for whatever reason, the periostracum will become damaged, and it cannot repair its periostracum after it's been formed. So I like to think of the periostracum much like the paint on your car. So the paint on your car protects the metal of your car from the elements outside. And the periostracum on the pteropod protects the shell from the water that's around it. So when the periostracum is intact, just like when the paint on your car is intact, the shell underneath can't get damaged. And it's really cool because this can prevent pteropods from being dissolved in water which is undersaturated with respect to aragonite. However, if the periostracum is chipped, just like if the paint on your car is chipped, the shell underneath is exposed to the water that's around it. So if it's exposed to undersaturated water, that shell is going to dissolve. And you can see in this image of the pteropod up here, there are white areas on the shell, white patches on the shell. And these patches are underneath areas where the periostracum has been damaged, and the shell is therefore exposed to the water. So it's doing the equivalent of rusting, it's corroding because of the undersaturated water that it's now exposed to. So I was interested, right, okay, if you can't repair the periostracum, you've got this organic layer on the outside of your shell, you can't repair it. But you expose the shell to undersaturated water so it dissolves, then you're going to get dissolution of the shell. I was interested, can a pteropod repair its shell? So I think we're going to do another poll at this point just to see what you guys think before we go through the data. Do you think a pteropod can repair its shell? So it's just a yes/no answer.
Right, Rosie, responses are coming in.
All right.
We'll leave the poll open for just a few more moments.
Yeah, that sounds good.
All right, we're going to go ahead and close it. And it looks like most of the folks online, 67%, think they can.
They can? Oh, so positive. Oh, that's good feedback. All right, well, we'll see. We will see what we can do. Thank you. Okay, so in order to test this question, we looked at some pteropods that were collected from the Arctic Ocean on the east coast of Greenland. And this is some work that I've done with collaborators at the British Antarctic Survey, primarily Vicky Peck, who is pictured in the top corner of the slide. And the interesting thing about this area is that for part of the year, the area is covered with permanent sea ice. Not permanent sea ice. For part of the year, the area is covered with sea ice. And so we took samples from these areas which had been under sea ice in the winter, and then in the summer, the pteropods were again exposed to, the water was exposed to the air again. So the sample that we're going to be looking at today is from this orange sample point here, which is in this red area, which means for 90% of the year, that it means that the ice was like 90% forged together when they took the sample, so pretty crazy. So this is just a diagram, a cartoon of the conditions that the pteropods would be exposed to through the year. So in the summer, the sea ice would be broken and the water could exchange freely with the atmosphere. Then during the winter, the thick sea ice forms. There's no exchange between the atmosphere and the water. So that means any breakdown of organic matter, that carbon dioxide that's released during that process is just going to accumulate in that water. So the water actually becomes undersaturated with respect to aragonite. The value is below 1, so it becomes chemically unstable for those shells. And then in the summer, when the sea ice breaks up again, there's more exchange with the surface and there's higher productivity in the surface ocean. And so then the water then becomes super saturated with respect to aragonite again. So it goes from about a 1.4 to just below 1 and then back to 1.4. So the pteropods are exposed to changing conditions throughout the year. So to do this, the pteropods were collected using a net called a bongo net. And this essentially is just a fishing net. There's 2 big rings at the top of it. Then the nets are suspended from that. And then there's kind of a bucket attached to the bottom of the net. And these nets are lowered down through the water column to about 200 meters. And then they're hauled slowly vertically through the water column. And the haul itself takes about 25 minutes because we don't want to pull the nets too quickly, because the pteropod shells are so fragile that even if you haul the net too fast, then you're going to break those shells of the pteropods. So once the net is back up, we take off the cod end, which is the name for this bucket, which is on the end of the net. We tip that into a larger bucket, and you can see in this example there was the little krill that was swimming around in there, and a lot of other stuff that doesn't really look like an awful lot, but it's a lot of algae that you find in these nets. And then from that, we pick out the pteropods and we use just a pipette like you would use in the lab. And we put them in these containers, which are like the soup containers that you get when you go for takeaway food. And then we looked at these pteropods under the microscope to have a look at what their shell condition looks like. And you can imagine this work is a little tricky because you're out at sea. So the boat is moving around and you're there on the microscope, which obviously is still, it's fixed to a table, but the pteropods are moving around in the water. So it's sometimes quite hard to actually see what you're looking at. But anyway, it's all good fun. So work on the ship. And we were interested in what these pteropods what their shells had been exposed to during the time they were underneath the sea ice. So this is a light microscope image here. This was actually taken when we were back off the ship. You can get much higher resolution light microscope images when the specimen isn't moving. And what you can see is that the central portion of the shell here has undergone considerable dissolution. And they just these little areas which have undergone dissolution. And then the outside of the shell, it looks absolutely fine. It looks kind of pristine. So I was interested, so these white areas are areas where dissolution has occurred. Just when the shell gets dissolved, it changes the way that light passes through the shell, so it always looks this white color. So I was interested, okay, has the shell thinned because the shell's been dissolved, or has it thickened because the shell is being repaired? And this is a really cool thing that CT scanning was in place to answer. And I know 67% of you thought that the pteropods could repair their shells. And the exciting thing was that they could. So, and CT scanning has let us see that. So the shell has not been fully repaired. So we'll just look at it in different regions. So first we'll look at this area here, which corresponds to this crack on, I suppose, the outermost crack on this pteropod. And you can see, so the diagram on the right, sorry, I should say, is a shell thickness map that was created from the CT scan data, where the thinnest shell is in red and the thickest shell is in green. So anything over 15 microns, 15 thousandths of a millimeter, so they're really thin, is in white. So that's thicker than 15 microns. So you can see that the majority of the shell is about that 8 micron kind of thickness. But in these areas where damage has occurred, there are areas where it's both thinner and thicker. So first, this area that we're looking at here, there's been considerable thickening associated with that area of dissolution, which suggests that repair is occurring. In these two regions down here, there's the area in the center of the damaged region looks like it's thin relative to the background. However, around the edge of that, there's considerable thickening of the shell. So it's almost like the pteropod is trying to repair that breach in its shell from the outside in. And this example up here is the repair looks almost further along. You know, it's had much more success repairing that, and there's now just a much smaller place where the pteropod shell is thinner. So this was really cool, but the exciting thing about CT data is that you can segment it so you can look at it from different angles. So we took a cross section from A to A prime, and this is the cross section here. So you can see that the majority of the shell is just a single shell wall. So it's just this really thin shell wall. However, in the two areas where we saw damage, so this area here, I don't know if you can see my mouse, but I suppose the coil to the left of the center, there's an area where it's just trying to repair underneath that bit where it's been damaged. And this area on the right here where it's done some considerable thickening of the shell underneath the damaged area. So let's zoom in on this area. And you can see here, so the average shell is about 8 microns thick, as we said previously. And this yellow dotted line is where we assume that the shell was prior to dissolution, just based on the shape of the other shell chambers. And you can see that the pteropod has added twice, or almost 3 times as much shell thickness underneath the area where the damage was occurring as it has on the rest of the shell that it's built. So it is really cool and shows that these pteropods can repair their shells. They do have a mechanism to prevent that water from impacting the insides of their shells. But remember, they can't repair the periostracum, so they can just repair these shells. But if they're in undersaturated water, the dissolution would still continue from the outside. So it's kind of a battle. So this is a kind of a schematic of what we thought happened to the pteropod that I've shown you pictures of today. So in the summer, it was swimming around in the ocean, and then there was a failed predation event. So this is a cartoon sea angel, like I showed you previously. And this failed predation event damaged the periostracum on the outside of the shell, and may even have caused a break in the shell, which you can see by this jagged line here. So then it was fine. There wouldn't have been any dissolution in the summer because the water was super saturated with respect to aragonite, so that shell was chemically stable. However, as we move into the winter, the water becomes undersaturated with respect to aragonite, and that shell will have started to dissolve. And then when you move back into the summer again, the water is again supersaturated with respect to aragonite. So the new growth of the pteropod that carried on after that, you can see the size of the pteropod has increased. The shell will have grown without a problem because it will have had fresh periostracum, and it's obviously supersaturated water, so that the damaged areas won't start dissolving anymore. So it's pretty cool. So we think the repair happened either in the winter or in the early summer. The other thing that you'll see that varies at the same time as aragonite saturation is food availability. And this is something that we don't really know the effect of this, but obviously in the summer, when the waters are supersaturated with respect to aragonite, there's also a lot more food because there's not that ice blocking the surface of the water. However, in the winter, the food's pretty scarce, and then in the summer, the food is available again. So this may also contribute to whether a pteropod can repair and grow its shell. So what did we learn? So we've learned that the periostracum, that organic layer which covers the shell, if that is breached or damaged and the water is undersaturated, those pteropod shells are going to dissolve in the areas where it's not covered, kind of like the rust on your car. But we found that the pteropods can repair their shells, which is really exciting, as it shows that they might be a bit more resilient to ocean acidification than we previously thought. They have this mechanism by which they can protect themselves from shell dissolution. What we don't know is what that costs them biologically. Like, are they less likely to reproduce if they're busy investing energy in fixing their shell? Or are they going to be less likely to escape from a predator? And we also don't know how long the pteropods can repair their shell for. Like, if there are just a few areas which are damaged, maybe you can repair it. You can see this guy here with just one bucket and many holes in his roof. You know, at what point can they not repair their shells anymore? At what point does the damage to the periostracum and the undersaturated water mean that their shells are going to fall to pieces? So we actually don't know that. And there's obviously, like I said previously, there's more questions than we have the answers to. So how can we use the data that we collected during this research to help you communicate about pteropods, their role in the food chain, and their potential risk from ocean acidification? Well, as I mentioned previously, pteropods are really small, about the size of a little grain of sugar. And so it's previously been really hard to talk to people about them because no one's seen them. No one's been to the beach and picked up a pteropod shell, or been swimming in the ocean and seen one swimming by because they're so tiny. And the advantage of CT scanning, which we really didn't see coming when we started doing this research, is that once you've done the CT scans of these shells, you can convert them into shape files and increase their size so that they can be used in science communication. So the shape files are actually just surface files. So you just create a file like a mesh of the surface of the shell. You can save them as a shapefile, and then we've been printing them between 50 and 100 times their actual size on a 3D printer. And I have a video here. So we just printed one on the MakerBot in our education department the other day. So this is not a particularly flashy 3D printer. I think if people have 3D printers, there's quite a lot of them. I did want to watch this print all day, but it took an hour just to print this one pteropod shell. So I have a few different pictures of it, but not all day. Oh dear. Okay. So at the end, you end up with this. At the end, you end up with this 3D pteropod. This one is probably about 4 centimeters across, which you can then use for education and outreach. So the massive advantage, obviously, is that people can now hold these specimens, which previously you couldn't hold. Like, I can crush a pteropod with a paintbrush. So when I'm doing research, I move them around with a paintbrush, and I've crushed more than I care to remember. And so you don't really want to hand them to a 7-year-old and say, look at this nice pteropod, because you know it won't exist by the end of it. So these 3D prints can be used for education and outreach, and you can pass them around and people can really look at them and understand. So the different pteropods that I have here, the one on the left is actually the one from the study that I just talked you through a few minutes ago. You can see, obviously this is not a super high quality printer that we've used, but you can see those areas where the dissolution had occurred on the shell. And then the center one is a Limacina retroversa from the North Sea. And then the one on the right is Helicanoides inflatus, which is from the Cariaco Basin. And we've used these in kind of a variety of events, and I've made there are a couple of videos online about how we've used this, which you're welcome to look more into. And I think Laura will post the links in the chat box on the webinar app. But the picture here is of me and my lab mates from Penn State, Ashley Gray and Heather Jones, at a K through 12 science fair in State College. And we 3D printed different types of plankton, and then we also had a microscope set up. And it was amazing once you showed the kids the 3D plankton model and then asked them to look for them under the microscope, they were much more likely to be able to pick out the different groups of plankton that you were showing them after they knew exactly what they were looking at. And they could look back at the 3D model and then back under the microscope. So it was actually a really cool tool to look at. So the plankton that we have here in blue, they're both pteropods, and then in orange, that's the foraminifera, which is a single-celled calcite-forming piece of plankton, which I also have 3D shape files of. Another thing that we did, Nick Holshoe and I co-developed a museum exhibit at Penn State University to get undergraduates excited about ocean acidification research, which was happening at the university. So this is the team that helped us build it. And you can see here on the left, we use these 3D prints in the exhibit and to help communicate about these organisms. We did have real ones under the light microscope, but just having them so big, and they are really pretty, people were excited to learn about the plankton. So the other thing is that this isn't expensive. You know, once you've done the CT scans, just to print them on the 3D printer, we actually managed to print them for free at Penn State, and I think there are quite a lot of education departments that have access to these MakerBot type printers. And actually, Laura, who's helping coordinate the webinar, has also used these shapefiles out in the Channel Islands National Parks for some education and outreach as well. So that's really cool. So how can you get these shapefiles? Well, I've made a Google form, which again, I think the link will get posted to, or we'll put it up at the end. So if you just fill in that Google form with your name and where you're working, just so I know where the shapefiles are off to, I can send you the link to a Google Drive folder which contains the shapefiles. And in there, I've got some information about the species names and where they came from. And I also have the foraminifera files in there as well. If you're interested in teaching about many different types of plankton, these are the ones that I've looked at so far. And then obviously feel free to email me with questions or to let me know how you're getting along. I'd love to know how these shapefiles are being used. And like I say, this is like been a really cool offshoot of the research that I've been doing on pteropods, is that we've been able to do all of this outreach stuff. So I'm really happy to help you guys. If you want to use the shapefiles in your classrooms or in your museums or wherever you are, just let me know how I can help you. So finally, I just want to quickly talk you guys through what I am interested in studying next, just so you know what's next on the cards for me, which questions I'm chasing, and how I'm going to keep using CT scanning to look at pteropods. So just to recap, you know, pteropods are at risk from ocean acidification because of their shells and where they live. And we found that on short timescales, so on the period of probably a year we were looking at in the previous experiment, they can repair their shells, which is so cool. But what about on longer timescales? What are the impacts in changing ocean conditions on pteropods on the timescales of 10 years, 50 years, 100 years? So can pteropods adapt or evolve to cope with these changing ocean conditions that they're going to be subjected to as we continue to change the Earth's climate? So one really cool way that people have looked at this in other species is to culture the organisms under projected future seawater conditions. So you get a big tank, you change the chemistry so it looks like it's going to in 2100, you pop your organisms in and you see what will happen. But sadly, pteropods are really hard to culture. So they feed using a giant mucus web, which you can see here on the image in the left. I've outlined it in orange so you can see it a little more clearly. But essentially, they emit this big mucus web and they catch all their favorite things from the water column, which are other little types of plankton, really. And then they ingest the whole mucus web and the things that they've caught in it. So this is similar to you flying along with a parachute, getting all your favorite snacks, and then eating all your snacks and the parachute. So the disadvantage of this is, as I said before, they're used to living in a big water column and migrating at 200 meters a day, whereas often your tank is much smaller than that. So they end up getting tangled in the bottom of the tank, wrapped up in their own mucus web, which is not particularly great. So they're not easy to culture. And the other negative thing about culturing is that you put these organisms straight into the conditions that they're going to experience in 100 years' time, which obviously isn't going to happen in real life. You know, these organisms are not suddenly going to wake up one day and the oceans change the chemistry that much. So we're going to take a different route to look at how pteropods have been impacted by changing ocean chemistry. And that's to look at how pteropods have changed over the last 150 years. So luckily for me, people have been passionate about pteropods for a long time. So these are two ships that sent out to do some of the earliest oceanographic expeditions, the HMS Challenger in 1872 and the USS Albatross in 1882. And they went out just to kind of see what is in the oceans, and they collected everything. And it's really, really well documented. Like when you look through the records, it's really impressive. So the people went out there in the 1880s to collect these specimens. And luckily, museums love keeping things that people have collected on these big expeditions. So luckily for me, I have been to the Natural History Museum in London and the National Museum of Natural History, the Smithsonian in Washington, DC. And I'm based at the Academy of Natural Sciences in Philadelphia. So I have access to all of these specimens that are in these museums. So probably when most of you have been to museums, you've seen the front of house or the dinosaur hall. This is our dinosaur hall here in Philadelphia. But what you probably haven't seen is that behind the scenes, there are big collections of materials. So this is the malacology or the snail collection at the Academy of Natural Sciences. And there are just cabinets and cabinets and cabinets full of snails. So this is what it looks like if you go down one row. So you just walk down the row until you find the label of the species that you're interested in. And then you walk down that row and you find the cupboard that those are in. And then you open it up and it's full of drawers. These are all full of specimens. So the Limacinidae, that's what I'm interested in. If you go in that drawer, it's full of other little vials which have these little labels with them and tell you a bit about where those samples were collected. So this is an example of what you get. So there's a little vial here full of pteropods, and these ones were taken off the coast of Scotland, actually, back in the, I think, 1847. We're really trying to get the dates confirmed. So you get these little labels, and they have the number at the top. That's the collection number. They have the species or what the species was called when it was collected, and then where it was collected. And from that, you can go and look in these big books called ledgers, where the collections manager has written everything. When they took that sample into their collection, they wrote down everything that they could find about that sample. So maybe they would write down the location that was taken from in the ocean or something. And yeah, so it's been really cool. It's like a combination of history and science at the same time. So this is what I've got so far. The plan is to build kind of a spatial and temporal record of how pteropods have been impacted by changes in ocean chemistry over time. So this is a map of the specimens I've got so far. It's color-coded every 50 years. So it's pre-1850 is in yellow. That's just that one sample off the north coast of Scotland. And then green is 1851 to 1900. Pale blue, 1901 to 1950. Darker blue is 1951 to 2000. And then the purple is 2000 to present. And as you can see, most of my samples cluster down the east coast of the US. So that's probably where we'll end up focusing this timescale study that we do. You might notice that some of the samples are currently on land. So that's just because they've said they got them from Greenland, and I've just typed in Greenland. So hopefully we'll get that cleared up over time. But this is what I'm going to be working on, using these historic records to understand pteropods through time. And again, I'm going to be using CT scanning to measure the thickness changes over that time period. So in summary, pteropods are planktonic mollusks which are at risk from ocean acidification because of their shells and where they live. But the CT scanning enables us to look at pteropods in 3 dimensions. We found that pteropods can repair their shells, which is really cool. We didn't know that before, which suggests that they might be a bit more resilient to ocean acidification, but we don't know what this costs them biologically, and that's going to be really important to find out. And CT scans are useful for more than just research, and you're welcome to print them out for yourself, and I look forward to hearing how you guys all use them. And with that, I'd like to thank my collaborators and my funding sources, and thank the NOAA Source Webinar coordinators for inviting me to give this talk. And obviously, thank all of you very much for listening. And I will take any questions that you have.
Thank you so much, Rosie. I can already tell you there's a lot of excitement on the line
Cool, okay.
And, you know, just not only about the research and, you know, applying it for science communication and outreach, but also that these tools are available. So I'm guessing people are going to be clicking those links. For those of you that didn't catch it, the links to get access to these files to print 3D is in the questions box. So you can find all that you need there to get access, along with Rosie's contact information. And so you'll also notice that we've got some great questions coming in. If you have any other questions, please go ahead and type those into the questions box. Anything we don't get to, we'll ask Rosie to follow up, and those will be stored in our archives, or you can, of course, contact her directly. Her email is in that chat box as well. So, Rosie, there was a variety of questions. How about we step back to your first time maybe seeing pteropods in the soil? And the question was, you know, why did, let me go back to it. So why did you know, why were the pteropods preserved in those sediments? Why didn't they dissolve there?
Yeah, that's a really good question, actually, and that's something that I'm also interested in. And I've done some decay experiments, so where I've taken a pteropod and killed it and let it rot for a few days and just see how those shells break down. Because yeah, chemically, if the water is undersaturated, those shells should dissolve. So the ones that I found were from the Cariaco Basin, which is off Venezuela. So that's a low latitude site, so it's quite saturated with respect to aragonite. So I think that might be why I found them. And also, if the sedimentation rate is quite high, like you can imagine, if there's a lot of stuff falling out of the water column, there just isn't, I suppose, the time for the shells to break down as they fall through the water column. So we did find them, and we found them pretty far back. So, and they've been found as far back as my colleagues just found some from the Paleocene-Eocene thermal maximum, which is 56 million years ago. And they just found the cast from the inside of the shell, not the shell itself. But at that time, obviously, the shells were getting to the fossil record. So it's a really good question, which we might not fully know the answer to at the moment. But there is this fossil record of pteropods.
Great, thanks. So stepping into the CT scanning, why don't you have to rotate the pteropod around a second axis to get that beautiful image?
Yeah, okay, yeah, good question. I know what you're thinking, because if you just had a camera, you could you'd have to rotate it on one and then a second axis. So because the X-rays will pass through the shell, they're not blocked by the shell, like the light would be blocked by the shell, but the X-rays can pass through the shell. So you can make a three-dimensional reconstruction of it, because you do actually image the whole shell in every image. But yeah, that's a really good question.
Great, thanks. So we'll have time for one more, and I'm going to combine a few here. So it goes back to, you know, that question of repair, and I think you hit on this a bit. But the question is, can pteropods repair themselves when they're living in those undersaturated waters, so omega under 1, or just in those supersaturated waters? And do you have any concepts sort of building on that? Is the repair from the outside, just due to the restoration of that saturation state to a value above 1, or is there some biomineralization process driven by the animal itself, which I know came in those further questions that you alluded to. So I don't know if you have an answer for that.
Yeah, so in terms of when the repair occurs, we actually don't know. So these specimens were sampled in the Northern Hemisphere summer. So by that point, they could have repaired during the winter, or they could have repaired in the early summer. We actually don't know that. And almost to do that, you would need to culture damaged pteropods and see if they could repair in undersaturated conditions. So people are working on culturing pteropods, and that would be a cool way to answer this. In terms of where the repair is occurring, I'm pretty sure that it's occurring from the inside of the shell, just because that external periostracum is damaged. And just by the shape of where the repair is, we see it kind of connecting the shell from the inside of the shell is kind of blocking that. So I'm pretty sure it's occurring from the inside of the shell. That's where the body of the animal is, and I assume that it will be able to create kind of a micro environment where it can increase precipitation of the shell.
Great. Thanks so much, Rosie. So we've come to the end of our time, and I know there are a lot of great questions. I think this might be the beginning. There are already ideas out there for ways to use these 3D images and print them for educational purposes. So as we said, we'll go ahead and any unanswered questions, Rosie will answer, and they'll be in that archive that will be sent to you via Google folder. In the next few days or so. And of course, you can contact Rosie directly with any questions or ideas or excitement. So thanks again, Rosie, for a really fantastic presentation. And thank you all for attending the Source webinar today. We do welcome any feedback, questions, suggestions for topics for the series, and we welcome that input. You can simply reply to the follow-up email you'll receive, or by contacting either myself or Laura. Our emails are listed on this slide. So you will also have the opportunity to provide feedback via a very short survey that will appear right at the end of today's webinar, and we'd really appreciate it if you could take a few moments to complete it, as that feedback is really what allows us to continue to host this series and inform NOAA of its impact. So as mentioned, we'll share the recording of this webinar via Google Drive, the resources that Rosie has mentioned and is sharing, along with answers to your question, and there will also be a video archive of this webinar on the NOAA Ocean Acidification Program's YouTube channel. So thanks again, Rosie. Thank you all for taking the time to join us. And this is the end of today's session.